สวัสดีค่ะ
สวัสดีครับ
Good morning and welcome to day 2 of Biotech 2021. This morning we will have sessions running in both rooms 1 and 2. The morning sessions for room 1 will consist of plant molecular research and plant protection, and the morning sessions for room 2 will consist of micropropagation and also physiology and production.
Before we get started with the oral presentation, we have a few remarks. First of all, a reminder that you can view our posters under the poster tab on the conference website.
Also, similar to yesterday, we will be showing each speaker's presentation in video format, and any speakers who are able to join us virtually will be able to answer questions in the joint Q&A session after the completion of multiple talks.
For this morning session, we will have a total of 5 talks before the joint Q&A session. Please feel free to type any question in the comment box at any time.
Finally, when you type your question, we ask that you please specify which speaker the question is for. All right, Natapong, I think we are ready to begin the session.
Okay, then let's get started. Welcome to the session 6, Plant Molecular Research. The first presenter of the day is J. Ramudu, who will give a presentation on DNA barcoding and molecular systematics in genus Coelogene Orchidaceae.
DNA barcoding and molecular systematics in genus Coelogene belongs to Orchidaceae. I am Dr. J. Ramudu, Assistant Professor in Botany, VSR Government Degree and PG College, Mumbai, Krishna District, Andhra Pradesh, India. Okay, so Orchidaceae is one of the largest families of the flowering plant. It comprises about 779 genera and 22,500 species distributed around the world. In India, 1,229 species spreading over 184 genera. It represents the second largest family of the flowering plants, and it contributes about 10% of Indian flora. The orchid genus Coelogene comprises over 200 species distributed throughout the southern East Asia, Sumatra and Himalayas. Most of the coelogenes are epiphytes. Some species are lithophytes or even terrestrial plants. Coelogene is with a total of approximately 550 species. Except a few publications of recent past, there is no work have been done on molecular characterization of Indian coelogenes. So I'm going to concentrate on this coelogene species belongs to Orchidaceae. So see, this is one of the coelogene species, that is Coelogene cristata, collected from southern India. Okay, so material methods. So totally I collected 9 species belongs to this Orchidaceae family. In that 8 species are coelogene species, 1 species poll data. So I collected the species belongs to southern India. So Coelogene brevis kappa, it is collected from Shaura Hills, Yarkad. So 2000 meters high altitude. So habitat host tree, it is epiphytic. The host tree is Alnus nepalensis. So like that, Coelogene corumbosa, Coelogene flaccida, Coelogene nervosa, Coelogene nitida, Coelogene vivalis, Coelogene prolifera, Coelogene stricta. These are the 8 species collected in southern India. So particularly southern hills, that is the Dodabetta, Ooty, Udakamandal, and also National Orchidarium Yarkad, and also National Orchidarium. So these are the sites I collected different coelogene species, and also include the Foliodata pallida. It is collected from Shaura Hills, Yarkad, Tamil Nadu. Okay, so these are the some these are the what I collected from different plant species of the coelogynes collected from southern India. Okay, so now, so see, this is the picture of some coelogyne species: Coelogyne brevis kappa, Coelogyne ovalis, and also some species like Coelogyne corumbosa, Coelogyne nitida. These are the plants I collected from southern Indian uh, areas, so particularly Shaura Hills, Yarkad, and also Orchidarium, and also Dodobetta Udakamandal. So these are some species I collected from different parts of the southern India. Okay, right. So coming to the material methods. So these are the material methods I collected the plant. For molecular studies, the plant materials, different populations, one population each belongings to the tribe Coelogynae, subtribe Coelogynae, subfamily Epidendroidae, a family Orchidaceae were collected from different geographical locations of southern India, particularly in Andhra Pradesh, Tamil Nadu, and Kerala. Some species were also procured from Tropical Botanical Garden and Research Institute TBGRI, Palod, Kerala, and National Orchidarium, Yarkad, Tamil Nadu. So now, how I isolate the that is isolation of genomic DNA from these plants. So the genomic DNA was isolated using CTAB technique, that is N-acetyl N trimethyl ammonium bromide technique, following the methodology Doyle and Diol 1987, was employed that extraction of that is the genomic DNA. So, and now I am going to this material methodology. Yes, so these are the some species which I collected from different southern regions of the India. Okay, right. So analysis, first amplification of RBCL and sequencing. So how I isolate the genome. After genomic DNA isolation from the plants, amplification of RBCL ribulose 1,5 bisphosphate, that is the enzyme, gene, and sequencing. So the PCR reaction mixture contained 1 unit of DNA polymerase, 2 ml of 10x PCR buffer with magnesium sulfate, 2 millimolar of each of the NTPs, 10 mm of forward and reverse primers, and 20 to 30 nanograms of the template of DNA. So this is the final volume was made to 20 ml with autoclaved MQ for amplification of the RBCL locus from the chloroplast genome. The thermocycle followed like one cycle of 5 minutes at 94 degrees Celsius, 35 cycles of 30 seconds each at 94 degrees Celsius. So this is the PCR reactions process. So after that, the PCR product was electrophorized in 1% TAE buffer agarose gel and containing 5 mg/ml Ethereum bromide and visualized on a UV trans illuminator. So the PCR products already are cleaned by exosap method. So after the cleaning of these PCR products, analysis of DNA sequencing data. How to analysis of DNA sequence data? So now the analysis of DNA sequencing data. That chromatograms obtained from the sequencer was base called using Fred. The sequences with the Fred score more than 20 were taken for further analysis. The forward and reverse sequences were trimmed and assembled using sequencer, and each sequential project file consisted of all the sequence of single species, and its consequences sequence was taken as the representative sequence for that particular species. So the representative sequence for each species was used for determining the interspecific K2P distance. So this interspecific, how to calculate the interspecific K2P distance? So the species identification success rates for each locus was determined using KTP distance arrived at on the basis of analysis of species utilized the Mega 4.0 software. And the second method is the neighboring joining tree. So for the distance, and third one is a blast method. So for the distance based method, species resolution of RBCL locus was calculated according to the following formula. So total number of species minus number of species with zero distance estimated by total number of species into 100. This is a formula to calculate the that is according to the distance. So how the interspecific K2B distance and species discrimination for RBCL at the tribe and subtribe level of coelogynes. The two genera, namely coelogyne and foliate of subtribe coelogyne, were studied here. The interspecific K2B distance matrix of 11 RBCL sequences obtained from 11 species of the ranging 0 to 0.017 out of species analyzed by and zero distance. The maximum interspecific K2B distance was recorded in between coelogyne trinervis and coelogyne flaccida. So species resolution. So species resolution can be calculated by the distance based method. The RBCL distance matrix of 11 species belongs to the subtribe coelogyne could not be resolved because of their zero distances with other species and thus resulted in a number of species pairs with the distance estimated as 0. So the consequently some species remaining unresolved. Therefore, the species resolution based on RBCL sequence is 36.36%. The species with the zero distance, that is closely related species, Coelogyne breviscapa, Coelogyne cristata, Coelogyne nervosa, Coelogyne nitida, Coelogyne vivalis, Coelogyne prolifera, and Foliata articulata. So average K2B distance score like this Coelogyne species: Coelogyne breviscapa, Coelogyne corymbeza, Coelogyne cristata, Coelogyne nervosa, Coelogyne nitida, Coelogyne prolifera, Coelogyne trinervis. This shows the close relative. So these are the nucleotide sequence in coelogynes, so different species. So allied nucleotide sequence of RBCL gene from the coelogynes. So phylogenetic tree building method. So the analysis of aligned RBCL sequences simplified from the species revealed that there are 144 variable sites, parsimony informative 17, singleton sites 29, conserved sites 861 out of total 1127 nucleotide sites. So the neighboring joining tree with 1,000 bootstraps replicates revealed 3 different clusters comprising 7 unresolved species, thus enabling species resolving of 72.72%. So the species clusters formed were shown in figure 3. So these clusters are follow. That is first cluster Coelogyne corymbosa, Coelogyne trinervis, Coelogyne nitida, Coelogyne prolifera, Coelogyne cristata. The second cluster is Coelogyne nervosa, Coelogyne breviscapa, Coelogyne vivalis, Coelogyne articulata, foliate articulata, foliate pallida, and Coelogyne flaccida. So the third method calculated this blast method. The RBCL locus, the investigated species in the blast analysis afford 60.15% of species resolution. The species discrimination rate based on the method, this method 51.29%. So these inter-specific K2P distances and species discrimination rates for the RBCL at the generic level. So the inter-specific variations and specific discrimination rates among these the congeneric species were also calculated individually for RBCL locus for 3 orchid genera. So in the genus Coelogyne, 9 species have been evaluated in the present investigation. These are Coelogyne breviscapa, Coelogyne corymbosa, Coelogyne cristata, Coelogyne flaccida, Coelogyne nervosa, Coelogyne nitida, Coelogyne vivalis, Coelogyne prolifera, and Coelogyne trinervis. The average interspecific K2B distance for RBCL is 0.07. So the RBCL locus had resulted in 3 species paired with the distance estimated 0. So the species discrimination rate in RBCL is 40.44%. Out of these 1180 nucleotide site of RBCL sequence of 9 coelogyne species. So okay, dendrogram of the tribe coelogyne based on the phylogenetic tree building method. So this dendrogram, we had to find out the two clusters. One is the Coelogen columbosa, Coelogen trinaris, Coelogen prolifera, Coelogen nitida, Coelogen cristata, and the second cluster is the Coelogen nervosa, Foldata pallida, Coelogen breviscapa, Coelogen volvalis, and Foldata articulata. So the Coelogen flaccida is outgroup in this figure. So when we are studied in this for discussion, so the RBCL locus from the chloroplast gene exhibited by the very low and varied species discriminatory powers using all three methods. The present study is based on previous investigation of the RBCL locus for reconstructing phylogenesis only at the family and subfamily level, and has its limited application at the species level. So interspecific variability and phylogenetic relationships from all the dendrograms constructed based on K2P distance bootstrap percentage, it is evident that the Coelogen flaccida belongs to Coelogen A. Its flaccida seems to be the outgroup. So based on that, we can construct the sectional delineation of the genus Coelogen lindren, arranged according to Joss and Chain, Subedian 2011. The section Ocellatae that belongs to the taxa Coelogen columbosa, Coelogen nitida. Section Cristatae, Coelogen cristata and Coelogen nervosa. Section Elatae, Coelogen stricta. Section Phalagenus, Coelogen vallis. Section Lentiginus, Coelogen breviscapa. Section Proliferae, taxa Coelogen prolifera. Coelogen flaccida section, that is Coelogen flaccida, and Coelogen trinaris. These are sectional delineation. So above the dendrograms are evident that Coelogen bovalis belongs to sectional phylogeny, shows the close affinity prolifera and cristata. The present Coelogen is polyphyletic. These results are according to the studies of Graham and Dale et al. 2001 and 2005. So the dendrogram shows that the Coelogen section oscillates under the which Coelogen columbosa and Coelogen nitida placed from the single cluster bootstraps with the other species, the cristata lentiginea. The present molecular results dendrogram indicate that there is a clear species discrimination of Coelogen nitida and Coelogen columbosa. So this is the dendrogram construction of subtribe Coelogen of the tribe Coelogenia based on the quantitative characters. So the present study also showed that there is a clear species discrimination of Foldata articulata and Foldata pallida, but low bootstrap percentage of 24 and 15 respectively. So invariably the genus Foldata shows the close affinity to the coelogines. This supports that the inclusion of the genus Foldata in the subtribe Coeloginae of the tribe Coelogenia. So and the conclude that the RBCL sequencing has proven an important tool in addressing the phylogenetic relationships at various taxonomic levels within the number of taxonomic groups. So molecular study shows that the Coelogen flaccida belongs to the Coelogen section. Flaccidae seems to be an outgroup. So the RBCL sequencing data also indicated that the Coelogen section oscillates shows close affinity with the cristatae and Antigoniaceae. So there is a clear discrimination, species discrimination on the Foldata articulata and Foldata pallida, but low bootstrap percentage of 24 and 15 respectively. Variable, the genus Foldata shows the close affinity with the coelogine. This supports that the inclusion of the genus Foldata in the subtribe Coelogine of the tribe Coeloginae. So also it's all the evidences that these are the sectional delineations of section Coeloginae. Thank you very much. Thank you for giving me this opportunity.
Hello, my name is Sachiko Isobe in Katsuta DNA Research Institute, Japan. Today, I'd like to present about our recent result of database construction named Plant Garden, which is a portal website for accessing plant genome, DNA marker, and SNP information. Before talking about Plant Garden, I briefly introduce our activities of plant genome sequencing in KDRI. We published the first plant genome sequences of Arabidopsis thaliana in 2000, then published many different plant genome sequencing by using diverse OpenGS technologies. In addition, we recently joined Earth Biogenome project, which aims genome sequencing all organisms on the Earth. Regarding its DB construction, we developed plant genome portal site named PGDBJ during 2011 to 2016. The database was for DNA marker and ortholog genes, and not start genome sequencing and gene information. Therefore, we decided to construct a new database, Plant Garden, which stores genome, gene, and SNP information, as well as DNA marker, QTL, and linkage map information. The name of Plant Garden is derived from Plant Genome and Resource Database entry. The beta version was released in 2019 March, and the regular version was opened in 2012 July. For the plant genome, several popular databases has already developed, such as Phytosome. However, the interface is sometimes difficult for non-genome sequence specialists. One of the reasons of developing Plant Garden is to provide plant genome sequences with a simple and user-friendly interface. The other aim is making a platform to provide both genome and DNA marker information. Plant Garden covering the plant species which are constructed high-quality genomes, as many as possible. This table shows the number of contents in Plant Garden. For example, we stored 158 genome sequences for 125 plant species. As you can see in this table, we also lots of contents for gene sequences, PCR-based DNA markers, and trait rating markers, and SNPs derived from SRA. From now on, I'd like to show the way of using Plant Garden. If you'd like to search the plant genome information from plant species, let's click Search by Species or All Plants. Then you can see the list of plant species in the database. And then you can select the target plant species from the list. After selecting the plant species, you go to Species pages. This is an example of species pages of Lotus japonicus. On these pages, you can access to publicly available genome sequences, gene sequences, and marker informations. SNPs on the publicly available SRA files are also analyzed by us and stored in Plant Garden. Genome and gene sequences are displayed for each version. The contents in PGDBJ, which is DNA markers, QTL, and linkage maps, are also stored in Plant Garden. They are also linked to the metabolomics and protein databases for each species. And you can link typical databases for each species as well. And when you select genome sequences, you go to the pages which describe assembled genome information. Here you can see sequences name and status of the sequences, such as number of sequences, total length, and number of genes. The data can be downloaded or seen by JBrowse. In these pages, you also be able to gene search and cut out genome sequences. Because genes are predicted on each assembled genomes, gene information is displayed for each assembled genome sequences version. And when you select gene sequences versions, you go to these pages. For example, if you want to know the whole list of gene sequences on the assembled genomes, let's put the assembled genome version's name here and click search. Then you can get the list of all gene sequences on the assembled genome sequences. These pages show the gene informations after selecting your target genes. In order to unify the annotation items and criteria of each gene in the databases, all genes are re-annotated with the annotation tools developed by us named Hayai Annotation. Gene search also can be done according to from gene functions such as GO and KEG based on the result by Hayai Annotation. Regarding as DNA marker, PCR-based markers such as SSR, CAPS, and SCAR are stored in Plant Garden. When you click the button, you can see the marker list and access to the marker details by clicking the marker names. Trait-related loci, which is usually called QTL, are also stored in Plant Garden. The traits are categorized in 6 classes: stress, fertility, yield, morphology or growth, quality or components, and others. The position of each loci are displayed on the map. You can read the details of QTLs by clicking the locus names on the list. SNPs information is also available in Plant Garden. This is a unique content of Plant Garden, and the variants were identified by us by mapping publicly available SRL onto the reference genome. The information are provided GVCF for each SRA and integrated VCF. Now we are considering that more user demands to compare the reference information in Plant Garden and use our own data. Therefore, we are developing tools that can seamlessly compare the data in Plant Garden as reference with the users. Several tools are already available, and you can see it by clicking Let's Analyze button. This is a list of tools which we currently provide: SNP detection and gene annotation tools named Hayai Annotation. KusagiDB is a database stored also of gene information from OrthoDB, Uniprot, and RefSeq for plant species, and used as reference databases when functional annotation is performed by Hayai Annotation. This is the final slide of my presentation and a message to the potential users of Plant Garden. First, Plant Garden is on the way of development. The database has not been perfect, and sometimes you feel inconvenienced due to problems or lack of functions. Please let us know if you have any problems or requests. Second, regarding the data to be stored, we would like to incorporate the opinion of users as much as possible. Please let us know your requests for data to be stored. We will respond to your requests as much as possible. Finally, we wish Plant Garden will contribute for the understanding and analyzing plant biodiversity. Thank you very much for your attention.
Hello everyone. I would like to present in the topic of molecular phylogeny and DNA barcode regions. Efficiency for identification the variety of Capsicum annuum in Thailand. Capsicum is a genus belong to family Solanaceae. 35 species is in native species of American tropical and temperate regions. Many species of this genus were distributed to Europe, Africa, and Asia through Portuguese traders about 300 years ago. These species, as Capsicum annuum, Capsicum sinensis, and Capsicum frutescens, are the greatest economic importance. For Capsicum annuum is in the most popular and important vegetables and spice, especially in Asian countries, and Thailand for a main ingredient in Thai food. Several varieties of Capsicum annuum were originated in Thailand, and some variety was planted in specific area. There are difficult for identification of this variety based on morphology. DNA barcoding based on coloplast DNA and nucleus DNA regions were tried to solve this problem. Previous study, the series of coloplast DNA, TINH, PHBA or TINF, TINT, and nucleotide form nucleus DNA as vaccine intron were used for identification of Capsicum annuum species in American sample. For ITS regions, show high efficiency for clear discrimination of Capsicum annuum varieties in Korea, whereas show the low genetic diversity in Japanese cultivars. In Thailand, genetic diversity among local Capsicum annuum variety has a few studies on the barcoding and did not clarify in the relationship. The objective of this study is to the phylogenetic relationship and genetic diversity among Thai Capsicum annuum variety and the test of DNA barcode efficiency for identify the variety of Capsicum annuum in Thailand. This is the scheme of this study followed by sample collection and taxonomic investigations. After that, study on molecular methods, phylogenetic analysis, and genetic diversity analysis. The plant materials was collected from various localities in Thailand and cultivated in greenhouse for investigate on morphological characters such as flower, fruit, and plant growth habitat. For molecular study, total genomic DNA was extracted by CTAP method and PCR amplification by using primer pair for ITS, RBCL, and MET-K regions and DNA sequencing. Phylogenetic analysis was separated as single DNA locus for ITS, RBCL, and MET-K. Each of single locus dataset was analyzed by using mast cell in MECAS7 program and calculated for the best nucleotide substitution models with J-model test. The phylogenetic relationship was performed by maximum likelihood and Bayesian inference with RAXML and Mr. Bay, respectively. The last one, phylogenetic tree, was visualized by FIT. Population nucleotide diversity index and haplotype data analysis of Thai Capsicum annuum based on ITS region was estimated by DNASP. and POT-ART program was used for generate the median joining hepatocyte network. The results show that 51 nucleotide DNA sequence and are successfully for PCR amplification and DNA sequencing from 3 DNA regions. The ITS region shows the highest number of nucleotide variability sites at 146 sites in total length of 645. Although RBCL and METK are longer than ITS at 714 base pair and 911 base pair, but very low number of nucleotide variable sites as 9 and 15 sites respectively. The phylogenetic tree of METK and RBCL shows the low genetic diversity and low efficiency for separate the capsicum annuum variety from other species that show in the mix of the grade of the capsicum other species with the capsicum annuum. In the other hand, molecular phylogeny based on ITS region shows the highest efficiency to identify species and confirm the Capsicum annuum as a monophyletic group, and the Thai variety was divided into 3 groups and shared morphological characters such as flower, fruit, and plant growth habitat. Between these groups, interestingly, the Bang Chang Ba variety showed high genetic variation and separated into 5 subgroups. This figure shows the maximum likelihood tree of Thai capsicum annuum variety based on ITS region that separated into 3 groups and did not relate to morphological character, which indicate by the tail of each sample in 6 characters. The first one, the row number 1, foot position. Row number 2 and row number 3 is the foot color of 2 states, young and mature. Flower position in row number 4. Anther and stigma stem color in row number 5. And plant growth habitat in row number 6. The Capsicum annuum Bang Chang variety in group 1 has high genetic diversity that separate into 5 lineages. Bang Chang number 6, Bang Chang number 3, and number 2, Bang Chang number 1, and Bang Chang number 5 is a strong rear of root support. White fidget variety has low genetic diversity and identical of morphological character in group 2. It is a fidget variety in group 2 that's the same morphological character. This figure shows the morphological character of Capsicum annuum Bang Chang variety as a fruitless color in mature, white to cream seed color, corolla white and without spot, and elyx of plant growth habitat. This result indicated that this variety as non-monophyletic group. To confirm the phylogenetic result, the population nucleotide diversity and hepatocyte network was analyzed. The Thai local varieties show high level of genetic variation, which indicated by the population nucleotide diversity index and hepatocyte network at 14 hepatocytes in 17 samples, 33 nucleotide variable sites, 35 nucleotide mutation and show the high number of the nucleotide polymorphism, hepatotype gene diversity, and average of number of nucleotide different. This table shows the total number of 33 nucleotide variable sites from 14 hepatides of Thai capsicum annuum variety based on ITS regions. The hepatide network of Thai capsicum annuum variety based on ITS sequence. Each color is related to previous group of phylogenetic results, and Bang Chang varieties still show high nucleotide diversity that were divided into 5 hepatocytes in hepatotype 1 and hepatotype 2, hepatotype 3, hepatotype 4, and hepatotype 5. The local Thai capsicum annuum variety had phenotypic character complex which did not relate to genotype. The ITS region was able to identify between capsicum species due to the nucleotide variability of nucleus DNA regions higher than the chloroplast DNA regions. The ITS region might be proposed for DNA barcoding to the capsicum annuum variety group in Thailand. Thai local capsicum annuum variety show higher genetic diversity than previous expect. Low genetic diversity of capsicum annuum was reported in domestic cultivar in Europe and Japan based on ITS region. Capsicum annuum cultivars in Europe and Japan revealed to closely genetic relationship and loss in genetic diversity due to invasive selective breeding. For the local capsicum annuum variety in Thailand might be reflect from genetic exchange during natural breeding and also not strictly in the breeding line selection by the local farmers. This phenomenon may support by the case of the domestic cultivar of capsicum annuum Bang Chang variety and Chifa variety that exhibit higher genetic diversity and variation than the breeding line of capsicum annuum Pichit variety. In conclusion, ITS region has potential as DNA barcoding for identification between species level and local variety in Thailand. These varieties show as species complex in phylogenetic relationships and unrelated with morphological characters. Hepatotype network and nucleotide diversity parameter indicated as the high genetic variation within Thai domestic cultivars. These results suggest that should be improved on plant genetic resource management in Thailand to protecting the loss of genetic diversity. In further study, should be investigated on chemotaxonomy and advanced molecular marker techniques to clarify the species complex within capsicum annuum cultivar in Thailand. And finally, we would like to thank the Thai Science Research and Innovation (TSRI) through Department of Agriculture for financial support. And thank you for your attention.
Good morning, honorable guests and ladies and gentlemen. Today I was really delighted to present about my study of sample preparations of the drying glangle in ethanol is affected to suppress Cavaliria species, which cause great discoloration disease or rights, this laboratory. So right is actually important step for cross worldwide, and more than 3 billion of world population is totally relying on the daily rice consumptions. And also in Thailand, right, is socio-economically important crop and an export product. X is the Thailand is the scam larger rice exporter in 2018. However, currently, right, had been faced with a lot of constraints. by the various biotech factors on both quality and quantity of the right production. Among those dirty panicles, we call the green discoloration disease one of the crucially important constraints right now. So what is the green discoloration disease or rags? Green discoloration disease or rags, we can call the green discoloration because it is a disease complex with multiple causal agents and particularly infect on right panicles with various necrodivisions of the right grains. And there have been conducted a lot of research. Green discoloration disease is caused by many causal pathogens, and also this is a major impact of the green discoloration. It can be effect on the seed potential and the seed quality and germination ability, and as well as the grain ability as well. And if the grain discoloration is infected, the main yield loss can be 80.9%, ranging from 0.1 to 60.2%. And some fungi are very deleterious because of they can produce some mycotoxin. It can be the cancer because it have the carcinogenic effect to the humans. Nowadays, to produce the food is totally relying on the chemical applications, for example, the chemical fertilizer or the chemical fungicides. And also in Thailand, a lot of farmers are using the chemical control measure in suppressing the grain discoloration. But actually, the chemical control have a lot of negative impacts to the environment and the human health, and also it can be developed in the pathogen population. So the applications of plant extracts and biocontrol agents are the potential alternative control measures in the future. So what is the Galanka? Galanka is the Zingiberaceae family, and also Appendix in tropical and subtropical Asia. It is used in now as a medicinal plant that has been used for the food and medicine, and currently it received a lot of interest in because of the ethanol ester that Galanka rhizome have acetoxy chavico acetates compound. It is very affected in controlling the Cavalierian species and Fusarian species. And also it can be easily accessible and available in almost household farmers in Thailand. So I was expected to classify the symptoms associated with the green discoloration, to identify the fungi which are associated with each symptom, and finally to determine the efficacy of the simple preparations of the drying calendar in suppressing the major fungi which cause the certain type of symptoms of the green discolorations in rice varieties of arboretum. So this is how I collected the rhizome calendar and how I prepare the drying calendar to get the drying calendar pounded in the laboratory. And also this is how I prepare the calendar solutions in 60% ethanol by using the maceration technique. So you can see, actually I use the ratios of 30 grams of dry calendar powder in 300 ml of 60% ethanol, and I left at the room temperature for 7 days, and after that the supernatant solution was decanted by using the clean cotton into water bodies. So now we have ready to use the Galanga ethanol extract. So this is the layout plant and the feed, how I divided the feed for the different control measure. So I use 10% dry calendar and 20% dry calendar, and also only 60% ethanol as a negative control, and also the net control without any control measure. And another is the pharmacoconjugation. So that is the mixtures of the chemicals and also the biofoliar sprays. Nowadays it is quite popular among the farmers. I sprayed only one time along the rice grain periods at the tillage stage. So this is how I collected the right sample. Actually, I centralized within each subplot by the area of 2 meters square. So we will have a four corner. So one corner represents for one replication. So I have totally 400 right panicle from one each subplot. And after that, I classified the green discoloration symptom as a small spot and small lesion, and also the lac lesion, and also the empty grain with the emphatic symptoms of the green discoloration, and even the heavy grain. So this just shows how I detected and isolated the cause of fungi or grain discoloration disease by standard blotter method. Actually, in this method, I was modified some implement. For example, like the use of a plastic cup instead of using petri dish, because it is quite easy and it is quite easy to incubate, and they didn't take a white space on the table, and also easy to handle for the scientists who are working in the laboratory. And after that, the incubation was left at room temperature for 5 to 7 days. So after 5 to 7 days, I was identified the fungi according to colony appearance and morphological characteristics. And finally, I calculated the percentage of the abundance of the microbiota associated with each type of JD symptoms, and I compare among the treatment by using the DMRT, using our program. So this is the fungi, the major fungi are identified from each type of symptoms. I identified not only by the molecular identification, but also by the morphological characterization. And totally, I have got Cavaleria lunida, Ordinaria ordinata, Bipolaris oryzae, and Fusarium oxysporians. The four major fungi were detected from each type of grain discoloration disease symptoms. So this is the percentage of the fungi associated with each type of grain discoloration symptom. According to the table, you can see clearly here Ordinaria ordinata and Cavaleria lunida play the most important role in every type of green discoloration symptoms and even in the gook seed. And this is the efficacy of the drying calango, ethanol extract in suppressing the Cavaleria species, and lactation in small spot, and as well as in the empty grain. By looking at here, only 20% calango show the significantly different in inhabiting of the Cavaleria lunata, Cavaleria species. So that's why we can be concluded that 20% galanka is as affected as the former conclusions. So there had been a lot of paper have been credited with the great discoloration diseases. And in 2017, Medin also studied with the each type of lesions of the green discoloration symptom. In our study, we also have the same lesion symptoms. But in our study, Alternaria, Alternata, and Cavaleria lunata are the most deleterious fungi in varieties of Amoritu. Even though another study have shown the Cavaleria lunida is the most highest infectious fungi, but in our study, the Ordinaria ordinata and Cavaleria lunida, these two fungi play most important role in infecting the green discoloration disease or rights, the varieties of Amoritu. So we can be concluded that in our study, in the right varieties of Arboretum, the symptoms of the green discoloration is composed of lac lesion, small spot, and also the empty grain with the emphatic lesions. Alternaria alternator, bipolar oryxil, Cavaleria lunita, and Fusarium oxysporans are the four major fungi from each type of symptoms. Among those, Alternaria alternata and Cavaleria lunita play the most important role in causing the empty grain in the right variety, Sabamori 2. So 20% Kalanka only can suppress in controlling the Cavalier alunita. So I can be summarized the Kalanka plant extract can be used to control green discoloration disease in the rice field because it can be easily accessible and it can be available in almost every tiny household farmers. So to control the green discoloration disease, using the simple preparations of the drying calendar extract would be the promising alternative control measure in controlling the green discoloration disease for environmental friendly agriculture. Thank you very much for your pay attention.
Sawadee Good morning, ladies and gentlemen. My name is Tithi Wim. I come from Myanmar. Presently, I am studying master degree on bioscience for sustainable agriculture, Faculty of Animal Science and Agricultural Technology at Silpakorn University, Phetchaburi IT campus, Thailand. Today, I am going to talk about my research project. The topic is in vitro screening of crude extract from plants against Pseudomonas saccharia and Carbolaria lunata and testing the efficacy of a selected crude extract or Alpinia galenga against grain discoloration and other rice disease in the field. So, first of all, I would like to introduce you about the background and problem of the rice crop production. As you know, rice is our staple food, main food, and economically and nutritionally food. So, it is essential to promote the yield of rice and the quality of the grain for global food security. However, the rice crop faced by the attack of the pest and disease and, according to the report of Muhabatra 2018, the average yield loss by pest and disease is 37%, and grain discoloration disease was considered as a minor disease at earlier time, but nowadays has become an economically important disease because this disease caused the rice crop both quantitative and qualitative loss. And the average yield loss by GD disease is 80.9%, reported by BITE 2020. And this disease was named as a dirty grain or black death and is a complex disease because by the attack of the several microorganisms such as bacteria, fungi, and virus, among them, fungi is the most infected one. In Thailand, Phetchaburi, two fungal pathogens, Pseudomonas saccharia and Carbolaria lunata are the prominent fungi that associated grain discoloration disease reported by METI 2017. By the impact of this disease, the rice panicle began affect grain, reduce grain weight and poor quality. So, to reduce and control this disease, most of the farmers still use and widely dependent on the chemical fungicide, but by long-term effect, its side effect may happen to human health problem and toxic effect to ecosystem and environment. So, as an alternative safe control method, nowadays the use of natural plant product, botanical pesticide is raising and some plant products such as neem, garlic wine are globally widely used as a green pesticide. So, I'm also wanted to observe the efficacy of some plant extract. So, the objective of the my research is the inhibitory activity of some plant extract to control Fusarium saccharia and Carbolaria lunata fungi on the laboratory and then to test the efficacy of a glenca crude extract in control of the GD disease and other rice disease in the field, Phetchaburi, Thailand, rice variety Phetchaburi 1. So, firstly, 8 kinds of plant materials such as lemongrass, macrolide, neem leaf, glenca rhizome were collected from Wahin, Fakrakirika and around Silpakorn University, Phetchaburi. After that, 50 kinds of plant extract, fresh and dry were prepared by maceration technique as shown in figure. Plant materials were prepared to get fresh database and dry powder and then soak into the 60% concentration of ethanol as a ratio 1:10 for 1 week. After 1 week, precipitation was filtered and ethanol was removed, and the remaining extract was mixed with the stearin water and 50 plant extract were resid and ready for further tests. So, antifungal assay of 50 plant extract were tested by food poisoning technique. At first, PDA media was prepared and then put into the test tube. After that, 50 plant extract were mixed with PDA media for concentration individually inside the test tube and then poured into the three plastic cut per test tube. And then a piece of Etichlorophantine, Fusarin Secret and Carbonaria lunata were cut by stearin corporal and then placed in the center of the plastic cut above the PDA media and incubated at 28 degrees centigrade for 4 to 5 days. When the mycelium growth of the fungi from control treatment reached to full growth, the diameter of the fungi colony from all treatment well matured diagonally and calculated percent inhibition. So, the result of the screening test revealed that dry Alpinia galanga extract had totally inhibited on the growth of Fusarian secret and followed by the dry peat or mangrove line 50% inhibition and followed by the dry leaf or clavurat 40.42 inhibition. At control treatment 60% ethanol show no inhibition. And then on the growth of Carbonaria lunata fungal pathogen, the results show dry Alpinia galanga highly inhibited, 78.33 inhibition and followed by dry leaf or clavula leaf, 49.17 inhibition and followed by fresh leaf or clavula leaf, 43.33 inhibition. At control treatment, no inhibition was found. So, in the screening test, Alpinia galanga was the most effective, and then the Alpinia galanga was selected and ethanol galanga extract was prepared for the feed test. The extract was prepared the same procedure as the previous screening test, except the removal of the ethanol step. And then the feed test was done 2 times at 2 glory season at Nyang district and Thailand district. When the rice plant reached 45 days old, the rice pea was partitioned into 5 plots. And then first time spraying was done at tillary state, 45 days old CLD, and followed by second time spraying when the rice plant reached to milky state. And then this slide shows data collection of narrow brown leaf spot disease, neplaque disease, and GT disease. So sample collection of narrow brown leaf spot disease was done when the rice plant reached to 90 days or after sowing. By walking across the rice field, as the N-shaped or zigzag pattern and 48 fret leaves were collected from 60 places, 1 place for 3 fret leaves, and then the severity of the disease checked consequently according to the 5 rating score, score 1 to score 5, very mild symptom to very severe symptom, on the by checking on the fresh leaf. And then the sample collection or GD disease and neplaque disease were done 110 days after sowing. 400 panicles were harvested from each subplot. from 4 specially separated places. One places for 100 panicle, and then the disease assessment or GD disease and nephrotic disease were done at the ISA laboratory room. GD disease assessment was done by taking 10 panicles from each 100 panicles and check the severity according to the ED standard evaluation disease symptom. Scale 1 to scale 9, and then nephrotic disease assessment was done by taking 100 panicles and check the distant lesion throughout the main axis of the pedicle branch, and the disease incidence was declared as a percentage. After analyzing the data, the result found that among the 5 plots, the plot that spray with 20% ethanol extract significantly control on the severity of the narrow boundary spot disease in both seasons, and then GD disease control didn't show significant effect. However, the 20% ethanol extract could reduce the severity of the disease when compared to other plots. And for the nephrotic disease, in the growing season 1 didn't show significant effect and could not control the severity of the disease. But in the growing season 2, 20% ethanol extract showed the significant effect and 10% ethanol extract as well could control the severity of the nephrotic disease. Let me move on discussion part. On the screening test, Alpinia glanca extract was most effective and show its high efficacy on control of two fungi because of its major compound acetylchabicol and its 1-acetoxy and phenolic group has synergistic effect and can destroy the cell membrane of the fungi. And then the other two extracts from mangrove and clavula leaf also show their moderate effect because of their chemical component, beta-pinene, limonene from mangrove and glycoside and flavonoid from clavula leaf have antifungal activity, and they can control a moderate amount on the growth of the two fungal pathogens. And on the feed test, 20% ethanol extract show its high potential on the control of narrow boundary spot disease, GD disease and nephrotic disease in both seasons, but in the control of nephrotic disease in the growing season 1 could not control because there was a continuous heavy rainfall and flash flooding in the growing season 1 in Myang district. So, high temperature and relative humidity promote the disease severity and the dispersal of the airborne spore Mechnopodicrucial that cause nephrotic disease. And then the farmer used the plant hormone, and this plant hormone also favor the disease severity and may occur there a lot of the empty grain. That's why in the growing season 1, 20% crude extract could not control the severity of the nephrotic disease. To sum up, from our result, dry Alpinia glanca extract show its high efficacy in the control of two fungi in the laboratory and nephrotic disease, narrow boundary spot disease, GD disease in the field condition also. So, for the sustainable rice grain production, Alpinia glanca extract could be considered as an alternative control method in controlling of rice fungal disease. The last, I'd like to acknowledge Thailand International Cooperation Agency (TICAD) for their funded support to my story, and I'd also like to acknowledge Faculty of Animal Science and Agricultural Technology, Silpakorn University, for providing laboratory material and teaching infrastructure. And also would like to acknowledge to my respectful advisor, Dr. Rashawan Mogot, and to my co-advisor, Associate Professor Ajamanat, for their kind teaching and kind contribution to my research. That's all for my presentation. Thank you for your attention.